Thank you very much. St. Jude is a special place. Our mission is to advance cures and means of prevention of pediatric catastrophic diseases through treatment and research. And no child is ever denied treatment based on a family's ability to pay. And basically, the reason that that can happen is that we have an incredibly active fundraising organization. So 75% of the funding for the institution comes from private donors. We treat about 5,000 patients in any year. We get about 600 new cancer patients, about 300 new hematology HIV patients per year. We have a comprehensive electronic health record and a multidisciplinary team approach to patient care. We also have more than 90% follow-up for all of our patients for at least 10 years. We have survivors that are into their 60s because they were treated when the hospital opened in 1962. So we have very well-defined phenotypes both during cancer treatment and after the patients survive. We started a preemptive clinical pharmacogenetic implementation protocol in 2011 with the goal to implement preemptive pharmacogenetic testing for all St. Jude patients. We provide clinical decision support for at least one drug before we will move that gene into the EHR for the patient. And once a gene moves into the EHR, we move it in for all past and all future patients enrolled on this study. That's actually one of the most difficult challenges that we've had. And we provide information freely to patients and to others. We have a very active website where all of our information is available. So why are we doing preemptive pharmacogenetic testing now and for children at St. Jude? Well, preemptive genotyping avoids that long turnaround time between the decision to prescribe a drug and getting a genetic test available to prescribers, and sometimes that's been too much of a speed bump for clinicians to overcome. The cost of genotyping for most actionable loci is very low. It costs about the same to do a panel as it does to test a single gene. So it makes sense to test at all actionable multiple loci, and that makes multigenic preemptive pharmacogenetic testing inexpensive. Everyone in the world is at a reasonably high risk to receive at least one high-risk pharmacogenetic drug, especially over a lifetime. So the chances that the genotyping data will be used is high. We have a culture of prescribing that's based on evidence. We have a good EHR that impacts most of the prescribing for all of our patients. And so we sort of felt a few years ago, if we can't do it, as people who are experts in therapeutics and who know about pharmacogenetics and who don't have to worry about these financial problems so much, how is anybody else going to be able to do preemptive genotyping? So we decided to take this on. So as I mentioned, use of pharmacogenetically high-risk drugs is relatively common. These are data from the general population in the United States, where by the time you reach the 40 to 64 years of age, over 50% receive at least one high-risk drug, and over 30% receive at least two high-risk drugs. Of course, that number is much smaller in the general pediatric population, but our pediatric population is sick, so that number is high. The children, of course, that we do this genotyping in are going to evolve into adults. So, of course, the idea here for the future is that we will genotype people, preferably when they're young. That's a lifelong genetic test result, and it'll be available for use throughout their lifetime as their risk of being prescribed high-risk drugs keeps going up. We did a little analysis of what percentage of our patients were taking a high-risk drug, and about 50% of patients were prescribed an actionable high-risk drug in any one-year period at St. Jude. So we felt like that was a high percentage. So the use of the high-risk drugs is high. How about the frequency of actionable genotypes? And we did the math for the first 12 CPEC actionable genes, and 99% of the population, whether white or black, is going to have at least one high-risk diplotype for one of these actionable drugs. So again, doing a panel of even just those 12 genes, you're going to find almost everybody with something actionable. In our own patients, 90% of patients have at least one high-risk genotype in their EHR, just with the first 9 genes that we've implemented, and again, about 90% of our patients have at least one high-risk genotype, and several have more than one. What are special considerations in children? Well, in general, children clear drugs better than adults, so they tolerate drugs in general much better than adults do. There are some incidental findings that are more likely to be discovered and actionable in children than adults, and the one that has affected us is Klinefelter's, where boys have an XY chromosome defect, and that's about 1 in 600, so we've definitely found that. And it is actionable, so you can use tutors, you may even use hormonal therapy before the child turns 18, so that's something to consider when you're doing this, because if you do any genes like G6PD on the X chromosome, you are going to find Klinefelter's. We felt informed consent was needed because the parents have to consent on behalf of the patients. And we set up our study so that the patients were not allowed to refuse an incidental finding on behalf of their child. So at the time we get consent, we say, we might find that your child has a disease like Klinefelter's. If we do, we are going to tell your physician, and your physician is going to talk to you. If that's unacceptable to you, then don't enroll on this study. And the consent has to be reobtained at the age of majority for children, and I'll show you some data on that in a second. More changes in the healthcare system are likely over the life of a child than the life of an elderly person, so there probably are some special challenges thinking about how do we make sure that these results are available lifelong for these patients. So the incidental findings I mentioned have been something that we've discussed in detail with our ethics and our IRB committee. Luckily, these actionable pharmacogenetic genes generally don't have health implications outside the use of medications. It's just the fact that if you look at the X chromosome, you are going to be able to find sex chromosome defects in children. So we decided that we would have to disclose an incidental finding to families if there was a very strong association with the disease risk, if it was not likely to be discovered via other means, and if it's actionable before the child is 18 years of age. After 18 years of age, the patient has the right to refuse the information for themselves, but the parents don't have the right to refuse that information for the child. So some examples of something that theoretically would be disclosable if, let's say, we tested a pharmacogene that found out that there was a crazy high risk of teenage onset of some terrible disease like colon cancer that's findable by diagnostic screening, you would have to disclose a result like that. And I gave you the example of Klinefelter's. So I took this photo, which is from a tongue-in-cheek website, because the line in pediatrics is always that you're supposed to pretend that children are not really adults, little adults. But as someone who works in pediatrics, basically 99% of the decisions that we make are based on pretending that children are little adults. And it works really well. Like I said, they have great clearance. And so really, sometimes the desire to treat children like they're such a different species from the rest of the planet is not really applicable. And pharmacogenetics works quite well in children. So those lack of special considerations in children are that they do become adults, all of our kids do, that many of our patients are already over 18 years of age. Biologically, as I mentioned, we can really treat them that way. The consent, we have to get assent as needed from a child. And those germline genomic variants do not vary with age. That's one of the beauties of working with genetics. And every CPEC guideline has a section addressing the applicability for children, and thus far all of them have had some applicability. So at St. Jude, we're really approaching this pharmacogenetic implementation on two fronts. One is that we co-founded CPEC with investigators at Stanford University in 2009. And at about the same time, we started writing our local implementation protocol, where our long-term goal is to do preemptive pharmacogenetic testing for every patient as standard of care. And eventually, we realized that what we really want to do is to implement every CPEC guideline at St. Jude via this PGen for Kids study. So why did we create CPEC? It was a little bit selfish that we created CPEC, because we realized if we wanted to implement pharmacogenetic testing and guide prescribing based on genetic tests, we needed to do it based on the best possible evidence that had been highly curated and looked at by the world's experts on each gene-drug pair. And once you go to all the trouble of surveying the world's experts on going through the evidence for their gene-drug pair, you might as well bring that together, publish it, and make it freely available to everybody else. So that's why we created CPEC. And an important assumption of the CPEC guidelines is that we don't deal with the issue of whether to test. That's often driven by financial issues, and of course those differ country by country. And CPEC guidelines we want to be applicable to everyone. We also want them to work in the setting in which preemptive genotyping is done, where sequencing is so cheap that all of us have our genomes done, maybe at birth or early in life. And the challenge for clinicians is not whether to order a genetic test. The challenge for clinicians is to decide which genetic variants are so strongly associated with drug effect that it would be wrong not to use that information to guide prescribing. And that's what we try to deal with CPEC guidelines. And we have people from all over the world that are members of CPEC. And every one of our gene-drug pairs results in a publication of a guideline. CPT has first right of refusal on publications of these guidelines, and I think there's something like 25 or 28 of them published now. And each guideline is posted on the CPEC website, and this is where you really need to go, because there's often updates after publication. And so the information is really only up to date on the CPEC website, and all of the tables that support each of these CPEC guidelines are maintained on the CPEC website. So again, we're developing these CPEC guidelines, and in this context, we're also trying to implement pharmacogenetics for our patients locally, and we're trying to implement every actionable gene-drug pair. The only exclusion criteria we have are patients who've received a prior allogeneic transplant because their blood DNA is not their own, and patients who haven't received a liver transplant because so many of our pharmacogenes are highly expressed in liver. This is a list of all of the CPEC gene-drug pairs that have guidelines so far, and highlighted in yellow are the gene-drug pairs that we've implemented locally at St. Jude. Why are we doing this via a research protocol rather than just saying this is best practice, let's just do this? And basically, we wanted to we have all of these results that are generated in a CLIA, which is, I don't know what the equivalent in Britain is, but the CUL? CML. Regulatorily compliant, so that it can be put into the medical record and used. So we're testing for many more genes than we're putting into the EHR. So we feel like we have to have consent because we're withholding results from patients. And we tell them that up front. There's a lot of data that are buried in that array that we're not putting into your EHR until we know how to provide CDS for at least one drug for that gene. And so for that, we need consent, how to handle the incidental findings. We need to ask for consent when the child turns 18. Before we implemented the study, we worked closely with St. Jude's Family Advisory Council, and they actually agreed to record a video with us with just questions and answers. Did they have any concerns? And this video has been downloaded a bunch of times. It's still available. We looked at it again, and we think it looks okay, except I guess I do look 8 years younger. But I think the content is still good. So the process is we get consent, we get a blood sample. They have their genotyping done. It used to be 230 genes. It's now it's many more than that on the PharmacoScan array. All the results get FTP'd into a secure research database, and then there's this selection process of which results go into the EHR, one gene at a time, only as we develop the clinical decision support and all of the other educational materials that are needed to roll out that gene-drug pair. So again, our genotyping is done by collaborators at a company called RPRD in Wisconsin. They now use the PharmacoScan array. Every gene result comes as one file per gene per patient into our research databases. We have this firewall, and before we upload the data into the EHR, we have not only the need to develop clinical decision support, but a lot of quality control. For example, are the results concordant with the self-declared sex of the patient? That is not always true, and that is also a way where we can diagnose a disease like Klinefelter's. And also at St. Jude, we do a lot of research and a lot of phenotyping and a lot of research genotyping, so we look to see if the clinical genotypes are consistent and concordant with whatever other data we have for those genes for that patient. That's a very labor-intensive process. There's multiple steps to go into before we implement a new gene-drug pair. Of course, we have to interpret the diplotypes, develop clinical consults and problem list entries. We have to build interruptive clinical decision support. These are all directly supported by what CPEC does. So all of this is a necessary step for everyone, and CPEC can help with this part of it. Locally, we update our formulary and drug policies as needed. We have a public website. We update educational materials. We build competencies for all of the clinicians, our pharmacists who are involved in the interpretation. We have a pharmacogenetics oversight committee that reports to the pharmacy and therapeutics committee that then reports to the medical executive committee of the institution. So every gene drug pair we implement goes through all of the medical staff. And of course, we share this information with others. This is a timeline from 2011 to the present day of the gene drug pairs that we've implemented. And I have used color coding to indicate that sometimes, so first we did TPMT and thiopurines, then we did CYP2D6 and codeine, and everywhere in gold is just adding more drugs to CYP2D6. Now, some people would be frightened to take this stepwise approach, and maybe you will not have to do this because the CDS will already be created. It is a little scary that back in 2012, we knew that there were CYP2D6 poor metabolizers or ultra-rapid metabolizers, and yet we were prescribing drugs like ondansetron and oxycodone without any clinical decision support to tell people what to do. And believe me, if you don't have clinical decision support, you are going to have prescribing mistakes, even though the result is in the EHR. Without interruptive clinical decision support, the best clinicians are going to miss the gene-drug interactions that are important. So we're now up to 9 genes and 22 drugs implemented, and our most recent was 2C19 and proton pump inhibitors. So again, using those 9 genes, the percent of patients that have an actionable genotype for every gene is listed here. You can see for genes like 2C19, it's incredibly high. So there's a huge percentage of patients that have a potentially actionable finding with CYP2C19, at least with the American ancestral population. So overall, I think we've enrolled just at right about 5,000 patients. 94% of patients have agreed to participate, so we do have a few that refuse to participate. 97% have reconsented when they reach 18 years of age. We ask them when they're enrolled, do you want every actionable pharmacogenetic test result to be sent to you, to your home, in a letter that you can share with any doctors when we do that? And 96% of patients say that they do. And as I've mentioned, only 2 incidental findings out of 5,000, so very low percentage of incidental findings. The demographics are listed here, and this reflects the ancestral population for our referral base at our hospital in Memphis, Tennessee. So a fair number of African ancestry patients participating. By clinic area, it's mostly cancer, but we do have about 20% non-malignant hematology and about 10% HIV patients. So getting from the genotype And this is what is from whatever variant of calling file that one has, from whatever genotyping or sequencing method is used, getting from genotype to alleles, the functions assigned to those alleles, translating those alleles into diplotypes, translating those diplotypes into phenotypes, interpreting the phenotypes, and then assigning actionability to the phenotypes for at least one drug. Those are the processes that we need to go through. And again, it's CPEC guideline tables that help with all of these processes. These are our local data. So in the first 4,000 patients, we had 207 different diplotypes observed for CYP2D6. TPMT is much simpler, the best behaved pharmacogene in the world. So you can see that there's many fewer diplotypes for TPMT, but of course these diplotypes don't mean anything to clinicians. They have to be translated into phenotypes. Another point I want to make is that when we're doing preemptive genotyping, we don't insist that every single result be interpretable for every patient, for every gene. That makes it feasible to do cost-wise. If we wanted 100% perfection on getting every CPEC variant from this screening test, it would cost more money than what we're spending. For example, for these first 9 implemented genes, 3% of the results come back with an indeterminate diplotype. We are not comfortable assigning a phenotype to those patients based on the calls. By gene, that's very low for TPMT, partly because it's a well-behaved gene and partly because we insist that we get that right for most patients because we act on it in such a high percentage of patients. SLC01B1, it's much higher because there's a high frequency of variants for which the function is just not well established and we're not willing to call the phenotype. And CYP2D6, of course, has about 5%, mostly because of copy number variation that we don't insist on getting right in every patient. If we needed to, for a patient that we absolutely had to use a CYP2D6 substrate, we would pursue further molecular tests. But as a screening maneuver, it's not cost effective to get that down in every single patient. This is what the output looks like from our PharmacoScan. They're formatted reports. This is, again, 1 gene per patient. This is for 2D6. The called diplotypes are listed as the star allele diplotypes, and then in detail you can see every single variant that was queried in this assay that led us to assign this particular set of alleles. We interpret that in a consult with passive clinical decision support and assign the phenotype of rapid metabolizer in that case. Here's that consult that's templated, and actually we create these using a database because every consult has a section for phenotype assignment, diplotype interpretation, phenotype interpretation, and likely medications that may be affected, prescribing recommendations that are quite generic, and then an educational link. So we have a database in the background to build all of these consults for every single genetic test result. We created a pharmacogenetics tab in our EHR where all of the results are gathered together in a non-date-dependent fashion. So this just keeps growing with multiple genes as we continue to add them on. And we have the high-risk phenotypes automatically populate the problem list for patients. Also, you have the ability to manually enter. If a patient does come in with a 23andMe result, we can enter it into this problem list, and we drive CDS off of the problem list entry. Here, this patient is a CYP2D6 poor metabolizer and a TPMT intermediate metabolizer. And if that patient is prescribed thiopurines, the prescriber will get an interruptive alert telling them to adjust their dose. So it's those interruptive alerts that are an important part of what we do. We have 2 different kinds of interruptive alerts. A pre-test situation where the genetic test hasn't yet been ordered, so you need to warn the clinician you're prescribing a high-risk drug and you need to order this test, or the post-test situation, which I just showed you. So here's a pre-test alert. We don't allow coding to be used in our pediatric population unless CYP2D6 genotype has been ordered. And so, of course, what this means is that CYP2D6 genotype has to be a discrete, computable, findable result in the EHR, preferably one that we all agree to use the same terminology worldwide so that EHR vendors can help us create rules to find the absence or presence of a test. Finding the presence is easier than finding the absence. We have created, using a Delphi process, some standardized terms for clinical pharmacogenetic phenotypes and genotypes, and we've worked with SNOMED to incorporate these standardized phenotype terms into their vocabulary so that instead of having a nonspecific code, which they used to have, thiopurine methyltransferase deficiency, they now use our standardized terms, which are important because intermediate metabolizers get 60% of this dose, and poor metabolizers get 5% of this dose. So again, these post-test alerts are going to fire when the patient has a high-risk genotype and they're prescribed a high-risk drug, and we give them specific advice on what alternatives to use, and they can always consult one of our clinical pharmacists for more information. We also incorporate some non-genetic information into these alerts. So in this example, for voriconazole, we also incorporate age of the patient and route of administration to tell the prescriber what to do. We also, of course, are getting a number of 2-gene alerts that are popping up. Now we have NED-T15 and TPMT for thiopurines. A couple of anecdotes I'll just go over quickly, and I know I want to leave some time for questions. We've added a disclaimer. Interpretation may change over time, so please consult with us if you want to know if evidence has changed for the interpretation. We estimate that 5% of consults over the last 8 years will have had to be updated with a reinterpretation of their diplotype. We use the the presence of genotypes as a QC measure for our institution. So if we prescribe thiopurines to someone that doesn't have TPMT genotype done, we consider that a failure. We have had that situation where we accidentally get blood samples after a bone marrow transplant. Unless that is well documented in the EHR, that's going to happen. We have to be able to manage if genotyping is done elsewhere in this date-independent tab. If we find a new high-risk result on a patient, as we move a gene into the results section for patients over time, we have a pharmacist do a medication reconciliation to see if they might have been dispensed a high-risk medication some time ago that we don't know about. And we have to reconcile phenotype and genotype for those genes where we know something, like TPMT and G6PD. We have this need to designate a possible high-risk status, as I was talking to someone earlier. If a patient has one allele that's known high risk and the other allele that's function unknown, they're at least an intermediate metabolizer. They're at least at a minimum risk. So you want them designated differently from just unknown, and you want to be able to alert what happens. We update policies for the whole institution. And minimizing duplicate orders is an issue. Again, that's why we have to use common terminology, or otherwise we're going to have the same gene test being ordered over and over again for patients over time. So I'll just quickly acknowledge my colleagues at CPEC who are at Stanford and at St. Jude, as well as our guideline volunteer authors, and all of my colleagues at St. Jude for our protocol. And thank you for your attention.